In this short series of videos, we're going to be learning about two variations that occur in the Calvin cycle as a result of a fairly severe limitation by the primary enzyme involved in the Calvin cycle, Rubisco, more technically referred to as ribulose bisphosphate, its target, carboxylase oxygenase, which are the two types of reactions that Rubisco is able to catalyze. Now, the intended reaction, the carboxylase action, refers to the fact that Rubisco's job is to take atmospheric CO2 internalized by the plant and add it to the target molecule ribulose 1,5-bisphosphate, and we can see that being done here. As a consequence, this very unstable 6-carbon molecule is formed, which subsequently breaks down into 2 molecules of 3-phosphoglycerate, or 3-PGA if you prefer. Now, for this reason, because this is the default method of the Calvin cycle's function, we refer to this as C3 carbon fixation, referring to the fact that PGA, or 3-PGA, is a 3-carbon molecule and is the direct result of the carbon fixing actions by Rubisco. Now, the problem, if we take a look at Rubisco's action, actually involves the similarities between carbon dioxide, the intended target, and oxygen, the byproduct of the photosynthesis reaction. If we take the chemical structures next to each other, we can see that these molecules are very, very similar. Both of them are symmetrical molecules with an identical distribution, a symmetrical distribution of lone pairs, and both molecules are completely nonpolar and also linear molecules that are small in size. As a consequence of this, oxygen actually has the ability to enter active sites within Rubisco, and this is where the ribulose bisphosphate carboxylase oxygenase, this is where the last part of Rubisco's name comes from. Unfortunately, the oxygenase part of its ability as an enzyme is actually unintended, because as we can see here, if Rubisco adds an oxygen to ribulose 1,5-bisphosphate, instead of carbon dioxide, we get this, which is an unintended product that results, instead of 2 3-PGA molecules, now we actually get 1 fewer 3-PGA molecules, and because of the very delicate balancing act that we have in Calvin cycle that requires a specific number of carbons at each individual step, this unintended product that we get as a result of Rubisco adding oxygen to RUBP instead of carbon dioxide can cause the entire Calvin cycle to break down to an imbalance of carbon. So whatever we need to do, we need to prevent this particular step from occurring as much as we possibly can. But the problems only become compounded when we actually look at the cell structure inside of the leaf where photosynthesis takes place. So if we look at all of the green cells that are found on the inside, or rather in the middle of the leaf, these are specialized cells that we call mesophyll cells, or mesophyll cells if you prefer. And again, phyll implying that they are rich in chlorophyll, hence their green color, and meso is a prefix that simply means in the middle. And again, this is where the primary photosynthesis reactions occur, hence why we refer to them as the center of photosynthesis activity. Now, photosynthesis cannot occur without the ability of carbon dioxide to get in, because then Rubisco would have no target to add onto RUBP, and then the Calvin cycle would entirely break down. So the regulation of CO2 absorption by the leaf is controlled by specialized structures that are found on the outside of the leaf called stomata. Stomata is actually the plural. The singular form of this word is simply stoma for short. And stomata have the simple job of either opening or closing in order to control the flow of oxygen out of the leaf and CO2 into the leaf. Now, the problem is, and the reason why this oxygenase reaction is such a problem, is that if you think about it, the more photosynthesis occurs, because we know that carbon dioxide is a reactant, that means that as photosynthesis activity increases, the concentration of carbon dioxide inside of the mesophyll cells where photosynthesis is actually occurring is going to decrease, because the more photosynthesis we do, the more carbon dioxide is fixed onto RUBP, thereby entering the Calvin cycle, but also the more the light reactions are occurring, the more oxygen is increasing. So all this while, as CO2 is decreasing, the oxygen produced as a waste product in the light-dependent reactions is going to be increasing constantly, and this becomes a very severe problem, because if the stomata close, that means that the leaf no longer has the ability to import CO2, and if carbon dioxide is no longer able to enter, that means that all of the oxygen produced by the light-dependent reactions is trapped inside the leaf. And with a lower concentration of carbon dioxide, that means that it is increasingly likely that Rubisco will add oxygen instead of carbon dioxide, thereby catalyzing the wrong reaction to occur. And in the next video, we are going to look at two different but functionally similar adaptations that plants use in order to overcome this specific problem in environments where plants need to frequently close their stomata as part of their survival mechanism.